Hey team, Jack here, and in a previous video I did a big deep dive into flow cytometry and how it's such an awesome technique for immune cell research. But today I wanted to take you through a clinical example just to show that it's really important for diagnosing disease progression, disease severity, and whether the treatment is working, using immune cell populations as an example. And it's for this reason that in the basement, it's often in the basement of most hospitals, you'll find a flow cytometry machine, as well as pathology departments and places here. They use flow cytometry because it is essential for evaluating things like immune cell populations, particularly peripheral immune cells. So the disease we're going to cover is HIV, human immunodeficiency virus. Now it's a lentivirus, and under the electron microscope, we can actually see it looks quite similar to another virus I've been talking a lot about, which is SARS-CoV-2. And it is in some ways. It does have that phospholipid membrane, which it stole from us, just like SARS-CoV-2 did. And it does have a whole bunch of proteins embedded and protruding from that membrane. And those proteins are also important in binding to host cell proteins and molecules to pull the genome into the cell, which is very similar to SARS-CoV-2. But it's actually very different for a number of reasons, which I'm going to go over in the next slide. Now, unlike SARS-CoV-2, which binds to ACE2, and ACE2 is expressed all throughout our body, so SARS-CoV-2 typically gets all throughout our body. Unlike that, the HIV is much more specific, and it seems to target just a subset of T cells, and that's the T helper cell. And if left untreated, the T helper cells can drop dramatically, almost to nothing, and leave those affected with a substantially compromised immune system, which opens them up to other diseases as well as opportunistic diseases, some of which don't normally infect humans but can once our immune system is compromised. And it's these other diseases that can often cause HIV to become fatal. Now, right now, we typically get 750,000 deaths a year. So it's a very serious and important disease that's affecting humanity. But that number was higher early in the pandemic, but we've developed really successful antiviral treatments for this, to the point that if you are getting proper medical care with proper HIV antivirals, your life expectancy is actually not too different whether or not you have HIV or not. Unfortunately, whether you're getting those antivirals and good healthcare largely depends on where you live in the world. So it is a real issue about inequality. Wealth inequality often leads to health inequality, which is a real problem. So here is the incidence all over the world, and so we can see that it is a very prevalent disease. It is affecting a lot of people, but sub-Saharan Africa is a real hotspot for this disease, and it affects a lot of people in sub-Saharan Africa. And a lot of this is to do with infrastructure issues, healthcare issues, sexual education issues, as well as misinformation. And so this information, I just mentioned that. So I really want to touch on this just purely because I think it's an important issue. Okay. So when people talk about alternative therapies that predominantly work through the placebo effect, a lot of people say, you know, what's the harm? Let's just let people take these alternative therapies and let people get the placebo effect. So I'm going to go over how there is a real harm to this. And so a brilliant article, and in fact, in a chapter, an entire book called Bad Science, I highly recommend you read it, written by Ben Goldacre, made several allegations against this person here, Matthias Rath. Now, Matthias Rath, he was described in the article as an alternative medicine mogul who essentially sold multivitamins as a treatment for almost all diseases, including HIV, according to the article. And I'll just give you one little quote from the article. So they describe a case in which a pregnant woman newly diagnosed with HIV was visited at home by Rath Health Foundation employees and convinced to stop taking her antiretroviral medication in favor of Rath's vitamins. She died 3 months later. Now, vitamins are important, but if you're getting a full diet with fruit and veg, you will have all the vitamins you need. And any study investigating additional benefits from taking high-dose vitamins has not found any. The article in the chapter in the book described that this man essentially and totally got the South African government on board with this, essentially convinced them that the antiretrovirals were causing HIV and that the vitamins would be an effective treatment against this issue, alleged in the article. Now, Ben Goldacre, when he published the book Bad Science, it didn't have this chapter in it because they were currently under litigation in the UK. Ben Goldacre successfully defended that after having to sell his house for the legal fees, and he won damages to cover his legal fees in that case. And so now the chapter is back in the book. So I highly recommend you go read the book Bad Science. It will completely change your interpretation of the alternative therapy market. And I actually really think as a comedian, an Australian comedian, really nicely summed it up. Alternative medicine are therapies that have either been proven not to work or not been proven to work. Do you know what they call alternative medicines that have been proven to work? They call it medicine. Medicine does not care where the treatment comes from. A good example is, two of the most common drugs used in the world were chemically originally derived from plants. So aspirin was originally derived from the bark of willow, and morphine was originally, its chemical origins arise from the opium plant. So the short of that is, you would typically say bark extract and plant extract sounds like an alternative therapy. Well, no, they've been proven to work as pain relievers or anti-inflammatories in the case of aspirin. And so they are medicine, right? Medicine does not care where the medicines come from. They just care whether it's passed a well-designed clinical trial, okay? So anyway, I think it is an important message to say that there is harm to alternative therapies, and there are homeopaths and stuff trying to treat HIV and stuff. So the HIV epidemic is also partly caused by an epidemic of misinformation, and that's so important as well. Right, so let's jump into the virus. So here's a mature HIV viral particle. Now, the proteins on the surface of this envelope recognize the CD4 receptor, right? Bing, the light should go off. CD4 is a receptor that's only found on T helper cells. In fact, another name for T helper cell is a CD4 positive cell. And that's because the CD4 molecule recognizes the MHC2 molecule. Now, if you remember that sort of diagram, you've got the MHC2 molecule coming from the macrophage, you've got the T cell receptor coming from the T cell, and then you have the CD4 molecule reaching around both of those to recognize the MHC2 molecule. And that should tell you that CD4 is quite a long protein sticking out from the membrane quite far. So the viral particle actually needs to get closer in order to fuse its membrane. And so what it does is it binds to another receptor, CCR5, which is a cytokine receptor, a chemokine receptor, right? So which is also found on T cells. That receptor is found on a number of other cells, but CD4 is only found on T helper cells. So that's why it's a T helper cell specific disease. So after it binds to that, it can now fuse with the membrane and push its genome in. Now, it has an RNA genome, which is a lot like SARS-CoV-2. So far, this looks a lot like SARS-CoV-2, really. Now, the RNA genome creates a protein called reverse transcriptase, which turns the RNA into DNA. And now this DNA is then inserted, thanks to more viral proteins, into the host genome. So the HIV virus actually genetically engineers us. It chops our DNA and inserts its RNA genome, which is now being converted into a DNA genome, into our genome. And this is why this disease is so hard to treat. If you got rid of all the RNA, all the protein, all the viral particles, every scrap you could find of that virus floating around in the cytosol or in the blood of that patient, say that drops to zero, within the genome of the patient is still the HIV genome, which can generate viral RNAs, which can generate viral proteins, which then can create the viral particle. And so that's why this is proving so hard to cure. We can treat it, but we can't cure it. Now, I will say there's some interesting preclinical research out there where they use CRISPR genetic engineering technology to go in and genetically engineer the mouse to remove that HIV genome. So perhaps in the future, we will develop some genetic engineering technology which can cut out the viral genome from the human genome, and then maybe we can push towards a cure. But other than that, we're sort of committing that HIV patients will continue to take the antiviral drugs for the rest of their lives. Now, the antiviral drugs do a number of things. They're sort of a polypharmacy approach. They target the virus in several stages of its life. So they can help inhibit fusion of the viral particle. They inhibit the reverse transcriptase, which turns RNA into DNA. They inhibit the enzymes that choke the host genome and insert the viral genome. We can also maybe attack the chemokine receptor CCR5. We can block that to help block viral fusion and so on and so forth. So it's a very complicated polypharmacy approach to attack this virus, but we never get rid of it out of the DNA. So it's still sitting there dormant. Flow cytometry. How can we use flow cytometry to assess the symptom development, aka H cell loss in HIV? So we can take their blood, excuse me. The first thing we're going to want to do is some of those classical gating strategies. So here we have side scatter, which is cell complexity. Here we have forward scatter, which is cell size. And what we want to take are these cells here. So these cells will be the granular sites, the ones which are larger and also have a higher side scatter because they've got more cell complexity. So we draw a circle around them. So now we're just getting non-granule cells, which is typically lymphocytes and monocytes, the non-granule cells, the main non-granule cells within the blood. These down here, these really small things, are red blood cells, so we definitely don't want those. So we draw a box around that, and only those will now go forward to the next graph, right? So this is how this gating works. We let those cells through the gates to be analyzed in the next graph. Now in this graph, we've still got forward scatter, but we might have a dead cell stain here. And that's because dead cells can complicate your analysis. Maybe they get non-specific binding of antibodies, etc. We just want to remove the dead cells. So these cells are high in a dead cell stain, like propydium iodide, for example. So we want to remove those. It also tells us if something's gone wrong in the process. Say we had 50% dead blood cells. Something has gone wrong. Did the blood sit too long on the shelf? Was there an error in the way they took the blood? Was there an error in the staining? So this is sort of a quality control as well. The next thing we want to do is isolate our T cells. So we're going to look for CD3 on the y-axis and CD45 on the x-axis. Now you remember CD3 is a co-receptor with the T cell receptor, so it helps activate the T cell receptor. CD45 inhibits the T cell receptor, so they're both markers of T cells. If you've got CD3 and CD45, those interact with the T cell receptor, so they're expressed on T cells. So we draw a box around here, and these are our pure T cells. And then the next step is to look for CD4 on the y-axis and CD8 on the x-axis. So these are our CD4 positive cells, which are our T helper cells, and these are our CD8 positive cells, which are our cytotoxic T cells, right? Now, you might ask, why don't we just jump to this? If only T helper cells have CD4 cells, why don't we just jump straight to a CD4 stain? Well, the World Health Organization has recommendations for following this procedure, and they recommend doing this step to purify your T cells because you might have some autofluorescence in the background, or you might have some non-specific binding and so on and so forth. So it's just a way of double checking. You don't want to you want to make your clinical assessment as robust as possible. So having safeties involved, like measuring is it a T cell twice, is a good strategy. So they recommend doing this T cell isolation and then looking for your CD4 and CD8 populations. So let's go through an example. Now this example is actually, it's testing antivirals, and it is testing antivirals in a mouse model. Now here's the problem, right? It's called human immunodeficiency virus because it binds to human CD4 and it binds to human CCR5. So it won't bind to a mouse CD4 or CCR5. So what do you do? Well, this was done in 2007, which was prior to the CRISPR revolution, and CRISPR made genetically engineering really easy. I'm sure you've heard of it before. It basically revolutionized the genetic engineering system. So genetic engineering before the CRISPR revolution was much harder. So what you would do now that we have CRISPR as tools, what we would do is probably cut out the mouse CD4 and insert a human CD4 into that mouse genome, cut out the mouse CCR5 and insert a human CCR5 into their genome. Or you could add them to the genome, so you're not removing the mouse version, to allow the mouse to operate normally. So that's what you would do. We call those humanized mice. When you insert human genes into a mouse to look at a human system, you humanize the mice through genetic engineering. This was pre that, so they went with a different strategy, which is a bit of an ethical minefield, but I'll just summarize what they did. They took a mouse that was immune compromised. Essentially, this mouse strain, it's called a skid mouse, has, they took this mouse variant that you can create called a skid mouse, that has almost no immune cells. And the benefit of that is it can't recognize foreign tissue, right? It doesn't have that self-foreign recognition system, so it won't reject tissue that isn't compatible with it, right? So normally if you were to take human tissue, for example, and insert it into a mouse, the mouse would reject that. Its immune system will attack that human tissue. But in these mice, because they have almost no immune system, they will not reject any human tissue put into it. So what they did was they took those mice and they injected fetal thymus tissue and fetal bone marrow tissue taken from human fetuses that were remained fetuses, did not survive. So they took that tissue, the fetal tissue and the fetal thymus tissue and the fetal bone marrow tissue, and inserted it into the mouse. Now, the bone marrow will produce T cells and will go into the thymus tissue to mature. And so now you've got mature T cells floating around the blood of these mice. Now, that is an ethical cost. I 100% admit doing animal research and human fetal research is ethically tumultuous. But let's think about this, right? HIV was a death sentence. It led to death very, very quickly when it first appeared. Now, with effective treatment, you can live almost as long a life and as healthy a life as an uninfected individual, right? That is a huge medical success that has reduced human suffering massively. So I feel like the ethical cost that was paid for these experiments is 100% worth it when you look at the quality of life people who are suffering with HIV experience. Just a side note, right? There was a study that showed in Australia, if we kept all our cats indoors, we would save the same number of animals used. We would save the lives, by keeping our cats indoors, we would save the lives of the same number of animals used in animal research in Australia in an entire year. So 365 days of scientific research in Australia, all the animals that we used in those experiments, we would save the lives of the same number if we kept our cats inside for 4 days. Okay? So and cats don't treat their prey humanely, right? So perhaps we should maybe refocus our ethical attacks and I think if we look at the food industry, it's a matter of hours. If we all stopped eating meat, we would save the same number of animals in the food industry in a matter of hours as we would in all of scientific research over a year. Okay, so I think we need to put this in context, and we need to talk about it in terms of good, right? The amount of good that has come out of these antiretroviral drugs is massive. The amount of good that comes from the farming industry or letting the farming industry or letting cats outside is probably less, but it's costing us way more animals. Okay, so I think for some reasons animal research ends up in the public's eye when it comes to this issue, but I don't actually think it's as a bigger issue. I don't think it should be the first thing we discuss I think there are many much easier avenues to improve animal welfare on the globe. And I do think the ethical cost is worth the ethical gain. Like the amount of human suffering we've prevented with these drugs is phenomenal. Anyway, so we've got these mice with T cells floating around the blood, and here's a flat facts plot. CD4 positive cells up here and CD3 positive. So these are our CD3 positive, CD4 positive cells. And when we infect them with HIV, we see that that population disappears. All the CD4 positive cells disappear. And so these are our cytotoxic T cells that are left over, right? So we've got these cytotoxic T cells, but we don't have any CD4, CD3 positive cells because HIV has infected them and killed them. So now when we give them the antiviral treatments in these mice, we can see the restoration of that T helper cell population. So these guys look almost as good as these guys in terms of their T helper cell population. So the antiviral treatments has worked fantastically. So this is how we can use flow cytometry both clinically and in research to track the severity, progression of HIV and whether the treatments are working or not. So that was just a shortcut, but there's lots of things we can do in diagnosis. We can look at leukemias. We can look at whether someone might have rampant inflammation with neutrophils. We can look at all sorts of cancers of the immune cells. There's mast cell cancers. There's eosinophil cancers that cause the overproduction of those cells. So we can do a huge number of things to track cell populations in flow cytometry in the clinic to track diseases, and HIV is just a really good example of that. Thank you.